hi everyone. So it's my great pleasure to introduce Christine Mézard. Christine is one of the leading researcher to work with plant meiosis. She's working in INRAE, the National Institute of Agronomy and Environment. So as she just told me, she started with, she has a medical doctor degree. She got in 1933, and after she moved in the University of Paris Sud to get a PhD. Christine is really an expert in recombination, and she did recombination during her study from E. coli, yeast, and after she moved to know how recombination is working in plants. So make a big jump because from E. coli to plants there is a lot of more complexity for analyzing recombination. So Christine really, during her career, revolutionized the way to look at plant recombination. With new ideas. She developed technique to look at pollen typing, that is, look at the rate of recombination along the chromosomes. And she discovered that there is also different rate along chromosome, but also between male and female meiosis, that what is known as heterochiasmy. And now she's going to present some of the newest results that she published in Nature Plant last year. And the title will be New Players Acting in Plant Meiosis. Thank you, Christine, for staying so late.
No, thank you. Thank you very much, Arno. You are really too kind with me, and it's a pleasure to be with you. Even if I'm not in Mexico, I would have liked to come to see you, but it's very nice to discuss with you our new results. So today I'm going to tell you our last story about the new players we found that act in plant meiosis. And the work I'm going to present, so sorry, I'm going to make the laser pointer. Yeah, the work I'm going to present today it's work done by two fantastic collaborators, Nathalie Vreelinck, an engineer in our lab, and Marion Pusch, who is starting a PhD. So meiosis, as you all know, is at the heart of the eukaryotic life cycle. It allows the halving of ploidy. And the ploidy is restored by fertilization. So in all eukaryotic that reproduce sexually from men, this man that you know, or yeast, it's the same process. And this process, this halving of ploidy, is obtained by the fact that there is one set of replication followed by two sets of chromosome segregation. So from this, in this cartoon, I represented a theoretical species with two pairs of chromosomes. After replication, you have these two pairs of chromosomes having each chromosome having two chromatids. And the first meiotic division is very special because in this first meiotic division, you segregate the homolog to opposite poles. And in the second meiotic division, it's more or less like a mitosis. You segregate the sister chromatids. And central to this first meiotic division, there is something very important. You have to obtain this structure in which the two the homolog chromosomes are linked to each other. They form what we call a bivalent. So you have one bivalent here and one bivalent here. And chromosomes are physically linked to each other, you know. And so that's bivalent. And this bivalent can be visualized by looking at male meiosis. For example, here in Arabidopsis thaliana, it's a species where you have 5 pairs of chromosomes. And you can see this bivalent here on the metaphase plate at the first meiotic division. You have these 5 pair of chromosomes here. And at the first meiotic division, these homologs are going to segregate to opposite poles, here and here. And at the second meiotic division, the sister chromatids are going to segregate. And you obtain 4 haploid cells, each having 1 chromatid of each chromosome. And very important to this physical link of this bivalent to form this, to stabilize this bivalent, is a formation of crossing over. The crossing over, which is an exchange between it's due to a homologous recombination event, and this homologous recombination event leads to the exchange of chromosome arms between homologs. And that's very important to stabilize the bivalent. And it's very important to stabilize the bivalent, but it's also very important to get diversity because you create new combination of genes. So here you have the example of what happens when you don't form the crossover. So on the top here, you have the 5 bivalents that form at the metaphase 1. They segregate, the homologous chromosome segregates at opposite poles at the first meiotic division, and then segregation of sister chromatids at meiosis 2. But when you look in a mutant in which the crossover are not formed, the bivalent are not formed neither. You have here the 10 chromosomes that are random in the cell, that they don't know how to segregate to opposite poles because they have not recognized each other. So they segregate randomly at first meiotic division. And then at the second meiotic division, you get aneuploidy in the gametes that are formed. Aneuploidy leading to sterility. So what are the actors that control these different steps of meiosis? We know that we have around 80 genes that are involved in meiosis. We know that 28 are specifically involved in meiosis, but there are 51 that are involved both in meiosis and in DNA repair in somatic cells. How do the genes have been identified? They have been identified using different screens. Forward screens, reverse screen, or suppressor screen. The most common one is a reverse screen. You know a candidate gene in another species, you get the homolog in your species, you look for a mutation in this gene. So in Arabidopsis, we look for tDNA insertion or a CRISPR mutation. And when you have your mutant, you grow your plants and you see if there is a meiotic defect linked to this candidate gene. And how, using this strategy, there are several, I give a few examples of genes that have been identified, like DMC1, REC8, MSH4, or MSH5. Forward screen was something which was very tedious, and because what happens is you grow a series of plants, mutagenize your plant by inserting randomly a tDNA, and after that you screen for sterility in this collection of plants. And when you have a sterility, you look if the sterility is due to a meiotic defect. So it's very tedious, but that has been very fruitful because that has been done by Mathilde Grelon in our lab. And she screened a collection of 25,000 plants, and she got many genes with this strategy. And she identified genes like SPO11, PRD1, and the series of genes involved in meiotic recombination. And suppressor screen is also has been also very fruitful. So you start from a plant having a sterility, mutagenize this plant, look for plants that have restored fertility, and map the mutation that has restored the fertility. And again, it has been very useful to identify a series of genes that were involved in the regulation of the number of recombination events. So that has been very nice, but the two strategies, forward screen and suppressor screen, have only identified gene that had a strong phenotype when they were mutated. So genes that were not recombining at all, so you had no bivalent form, or a lot less bivalent, or when there was a strong DNA repair phenotype and chromosomes were completely fragmented. Reverse screen, you could identify mutations that were giving more subtle phenotype. The problem is that we know that we miss some players that are involved in the meiotic process. We know that we miss some regulators that would give more subtle phenotype. But why do we know that we miss some genes? And I'm going to give you an example, the example of the synaptonemal complex. Oh, I've lost my pointer again, it's here. So I told you about bivalents. Bivalent are here, but in fact, in the cells, the bivalent are linked by a proteic structure that glues the two homologs together. And this structure is called the synaptonemal complex. So here are cartoons that show this synaptonemal, the structure, the putative structure of the synaptonemal complex. It's a tripartite structure with two lateral elements and transverse filament, it's kind of zipper that zip the two chromosomes together. So if you look at this frontal cut of this synaptonemal complex, you have here the two chromatids, here and here. At the basis of this chromatid, the lateral elements like that, and in the center, the zipper that's formed by a protein that makes this transverse filament. This protein is called ZIP for zipper. And the synaptonemal complex is a structure. Oh, I've lost my pointer again. I'm here. So the synaptonemal complex is a structure that is very conserved in most eukaryotes. So it has been looked at by electron microscopy, and you see in species as diverse as Arabidopsis or a fungi, Sordaria macrospora, or Drosophila, or C. elegans, or mice, you have this tripartite structure, lateral element and zip in the middle, lateral element and zip in the middle. So you can recognize this structure in all these species. So it's very conserved. However, I don't know why I'm losing my pointer each time. So, however, the protein that makes this structure have no sequence similarities. So it's very difficult to find the protein from one species to another one just by looking at the protein sequence. So you cannot find, for example, if you look at the sequence, the sequence of the SCEP1 protein and try to find the homologs, you won't find it. For the lateral element, it's a little bit more conserved, but it's very hard as well to find the homologs of this protein from one species to another one just by the sequence structure. And there are proteins that are known to be at the center of this zipper-like structure here. And this structure here is called the central element of the synaptonemal complex. For central element, there are a series of proteins that are known in the yeast or in the worm or in Drosophila or in mice, but no protein have been identified in Arabidopsis thaliana. And there is no way to find this protein just by sequence similarities. And people, when they try to find this protein, they found them by screening with different kind of screens. So what is the role of this central element structure? The role is to help the polymerization of the transverse filament, stabilize the transverse filament, and reorganize the transverse filament. Because this structure that looks a bit, I present it as a cartoon, very stabilized, is in fact very fluidic. It moves, and protein are moving in this structure a lot. So the central element could be something that would stabilize this structure. When these central element protein are absent, the phenotype are different from species to species. In the worm or mice or Drosophila, it gives severe meiotic defect and sterility. However, in the yeast, Cerevisiae, it just delays the sporulation, and there is a slight reduction of spore viability. So it's not a very strong phenotype. So we wanted to see if we could identify this central element protein in Arabidopsis thaliana, and if we had this protein like that, what could be their role? Would that give a strong phenotype, or would that give a very a minor phenotype? So, as I told you, in the lab we do many screen, and we decided to set up a new screen based on transcriptomic data. When this paper appeared, we realized that the data they had on the transcriptomic were absolutely fantastic. They dissected Arabidopsis in 52 different organs or stages of development. And here, for example, for the flower, they had at least 10 different stages of flower development. And we were very interested in this stage because the size of this bud is exactly the one that we choose when we want to look at meiosis in Arabidopsis. It's 1.5 millimeters, and that's the stage of meiosis. And here, for example, in this data, you see that they could identify genes that were expressed only at this stage. So we look at their data, and we look at the profile of expression that we had for meiotic-specific genes. And so here you have the expression of the different genes, and here different stage of bud development, so from floral meristem to old flowers, and here we put as a control the root apex, because in the root apex there are many divisions, and we wanted to see if the genes were actively expressed in the apex, in somatic cell division. So that we got this profile for the 28 genes that we had that we know that were only involved in meiosis. We could see that they were peaking exactly at the bud size 0.5 millimeters. Most of them were almost not expressed in root apex, or after that the expression was out in older flowers. So that with having this profile of expression, we look at the profile of expression of genes that were involved both in meiosis and in somatic cells. And we know that they had a role during meiosis, but we know that they were also had a role in somatic cells, and that's the expression we got. And you see that the profile is less clear than the one that we had for meiotic-specific genes. So we decided to go to screen for genes in Arabidopsis that had a profile of expression like the one that we have here on the left. So we looked at the 25,000 genes in Arabidopsis, and we found 250 genes with a pattern of expression looking like meiosis-specific genes. And 250 was a bit too much for us to screen, so we decided to restrain our candidate genes between two limits. SCEP2 is a gene that is the less expressed during in the data we had, and DMC1 is the gene that is the most expressed in our data. So that gave us 80 genes in our list. It doesn't mean that the other genes are not involved in meiosis, but just that we didn't have the force and the people to screen more genes. We still have the list, and we may go back to this later. So what are these lists? So we have these 80 genes here, and what we decided to do is to look at mutation in these genes. So we decided to choose 2 DNA insertion lines per gene, and to search for meiotic defects by DAPI staining on spread myocytes. So we wanted to look at the chromosome behavior and see if things were going well or not during meiosis. And immediately after we got our list, there was this paper out in the literature, paper by the Jan Henderson lab, and he identified new genes involved in meiosis, which is a gene that is associated, it's a transcription factor. And when this gene is mutated, you see that it could have less crossover in one accession compared to another one. And we were very pleased because that validated our list. But this one, we abandoned the study of this gene. But what struck me immediately when I had this list of genes was this one, this gene here. Why? Because it was annotated as being a tropomyosin. And that's where my MD studies were interesting, because I know that tropomyosin interacts with myosin in muscle cells. I did a specialty in medicine, sports and medicine. So I was interested by muscle, and I know that tropomyosin is one interactor of myosin. And why is it interesting? It's interesting because the SCEP1 protein, which is the protein that makes the transverse filament in the synaptonemal complex, you know, the one that makes the zipper between the homologous chromosome, is annotated as being a myosin-like. So immediately I looked at this gene. That was my top candidate. And unfortunately, we didn't get any tDNA in these genes. And but I was convincing. I succeeded in convincing Natalie to try to knock out a tropomyosin gene in Arabidopsis. And she did the CRISPR-Cas9 on this gene, and she got a series of allelic mutants, 7 mutants here. And we looked at fertility, and we had a slight fertility defect when we mutated this gene. So that was interesting, and we went further and we looked what was going on in meiosis. So instead of having always this 5 bivalent, as in the wild type here, 100% of the cell, at metaphase 1, we could see that we had sometimes, in 10 to 20% of the cell, we had sometimes 1 pair of univalent. And that's something you never, ever see in the wild type. So that was interesting. And the consequence of having 1 pair of univalent, these 2 chromosomes segregate randomly at the first meiotic division, and sometimes they segregate in the same pole. So you have at the 6 chromosome on one side and 4 on the other side, on the other one, and that leads to that the reason why we have a slight fertility defect. So we call this protein, after we call this protein SCEP1, and that when AlphaFold started. So, and we looked at the structure of this protein by AlphaFold, and we found that it was a small coiled-coil protein. And we looked at all these other proteins here, and we found that most of them were either coiled-coiled or along alpha helix. So we were even more interested by this protein. And what is known with this, with the central element protein, is that they can form subcomplexes, like here with 2 protein of mice, or here with 2 protein of yeast. They make subcomplexes. So we looked in our set of protein if we had other coil-coil protein or alpha helix protein, and we got a series of protein like that. At this time, we asked a structural biologist, Raphaël Guerroy, And we asked him if one of our candidates coiled-coil protein could interact with SCEP1. And he told us, yes, indeed, one of your protein is an interactant of your SCEP1 protein. We called it SCEP2. It's not only an interactant, but the both proteins have co-evolved. So he told us they form a heterodimer. So we looked immediately at what was going on in this SCEP2 protein. So as in the SCEP1, there is a slight fertility defect in SCEP2, and as in SCEP1, in 10 to 20% of the cell, we have univalence at the first meiotic division and unbalanced segregation at the second division. So we have exactly the same phenotype in SCEP1 and in SCEP2. So we wanted to know if we had a synaptonemal complex form in these two proteins. And so for that, we use immunocytology. So we use antibody directed against the lateral element here called AZ1. And we use antibody directed against the ZIP1 protein, the one that makes the zipper here, ZIP1. So in the wild type, at the time where the chromosome synapse, the AZ1 signal faints and we don't see it very well. You can see it's very clear here, but that's a place where ZIP1 is not loaded, but it's faint a bit here, and that's where ZIP1 is loaded. So we can see very well the ZIP1 loading in the wild type. But in our two mutants, we see a strong AZ1 phenotype, and we never see the ZIP1. So the ZIP1 is not loaded in our two mutants. The transverse filaments are not formed. And when we looked in a high resolution, what was going on, we could see that two lines like that with AZ1, meaning that the two lateral elements were close to each other, like that. But they were not synapses. So we had alignments of the chromosome, but no synapses. So we raised antibodies against SCEP1 and SCEP2, and we looked at the loading of this protein, and we could see that the loading was exactly the same than the one of ZIP1. You look here, you have the SCEP1 signal and the ZIP1 signal and the merge, and they're absolutely colocalized. That's the same with SCEP2. So we have a perfect colocalization of SCEP1 and ZIP1. So SCEP1 and ZIP1 are on the synaptonemal complex. But now with high resolution microscopy, we can go in more details about what's going on. So when you look at the ZIP1 protein, we have antibodies that only label the N-terminal part or the C-terminal part. And we know that the C-terminal part is close to the lateral element, and the N-terminal part is at the center of the synaptonemal complex. So here in this study, we use RECADE, which is another protein in the lateral element. It's not ASY1, but it's RECADE, but they ASY1 and RECADE colocalize. So when we have an antibody directed against the lateral element, RECADE, and antibody directed against the SCEP2, you see that SCEP2 is between the lateral element. So you have lateral element here, and SCEP2 is in the middle. If we do the same with SCEP1, lateral element, and SCEP1 in the middle. So SCEP1 is in between the two lateral elements. But we can go further with antibodies directed against the C-terminal part. We see that here, SCEP2 is in between the C-terminal part. So SCEP2 is really in the middle of the synaptonemal complex. And we could also show that SCEP1 and SCEP2 colocalized together. So we have two proteins that co-localize in the middle of the synaptonemal complex. We obtained these very nice pictures by collaboration with Raphael Mercier at the Max Planck in Germany. So we now know that SCEP1 and SCEP2 are the first central element protein identified in plants and delocalized in the middle here. So, and what was very striking as well is that I told you that SCEP1 is not loaded in SCEP1 and SCEP2 mutants, but when we look at SCEP1, SCEP1 is not loaded in SCEP2 and ZIP1 mutants, and SCEP2 is not loaded in SCEP1 and ZIP1. So the three proteins seem to behave exactly the same. So we know, I mean, Raphael Mercier and James Higgins had shown that SCEP1 play a role in crossover formation. So we wanted to see if our protein was playing the same with crossover. You know, the laboratory of Raphael Mercier and the one of James Higgins published in back-to-back papers that the crossover in ZIP1 mutants, when the synaptonemal complex, the transverse filament are not formed, the number of crossover was higher. There was a loss of interference and the loss of heterochiasmy. So what is interference? Interference is a phenomenon that has been identified more than 60 years ago in the first genetic map that were obtained. And that was that's a fact that when a crossover is formed in one part of the chromosome, it inhibits the formation of another crossover nearby. So that's called interference. And interference is frequent in many species, and it exists in Arabidopsis. But when the synaptonemal complex is not formed, there is a loss of interference. And what's striking is that there is also a loss of heterochiasmy. In Arabidopsis, there are more crossover in male meiosis than in female. But if ZIP1, that is, if the synaptonemal complex is not there, the number of crossover is higher in both male and female meiosis. So what happens in our mutants? So we look at SCEP1. We look at first by cytology at the number of MLH1 foci. What is MLH1? MLH1 is a protein that binds to crossover. So it makes foci that you can count after. So in the wild type, there are around 10 MLH1 foci per meiosis. And in SCEP1 or SCEP2, there are around 50% more MLH1 foci in male meiosis. That's similar when you have the double mutant. And we performed a genetic map, and we confirmed this increase in crossover by looking at the number of crossover per myocytes. So we could look in female and male meiosis. In the wild type, you have around 2.8 crossover in the female meiosis and 5.4 in male. And in ZIP1, as I told you, in both sexes, crossover are increased, and there is no more difference in female and male meiosis. Both are around 7. In SCEP1, we had this increase in the number of crossover, 7.5 and 6.6, but there is a little bit of heterochiasmy. Heterochiasmy is not completely abolished. So what happens to interference? For interference, we have a ratio, and when this ratio is around 1, it means that there is no interference, and when this ratio is around 0, it means that interference is total. And in the wild type, what's going on is that there is interference in close by intervals. These are interval along the distance between two intervals along the chromosomes. So when you have two very close by intervals, you have interference. You don't find crossover in two close intervals. But when intervals are far away, you can find a crossover. But in SCEP1, the ratio is always 1, meaning that there is no interference at all. You can have close by crossover or far away crossover. And Marion looked at, we wanted to know if the genes, the SCEP1 and SCEP2 we had found in Arabidopsis were found in other species. And she could find SCEP1 and SCEP2 in all clade of angiosperms. But we started to have difficulty to find them in gymnosperms. In gymnosperm, here, for example, we have ZIP1, but we couldn't find SCEP1 and SCEP2. In Selaginella, we find the 3. And when we looked at more distant, it's very difficult. Sometimes we could have a hint, but we are not convinced because the size of the protein is very different. And what she could show as well, that not only we had homologs, but these homologs form dimers by alpha fold, as predicted in Arabidopsis. So what I told you about these two genes is that when we have mutation in these two genes, we have reduction of fertility, that the loading of ZIP1, SCEP1, and SCEP2 depends on each other. There is an increase in CO number, abolition of interference, and reduction of heterochiasmy. So it seems that SCEP1, SCEP2, and ZIP1 form a functional triad. I told you that we found that SCEP1 and SCEP2 colocalize in the center of the synaptonemal complex. So they are the first central element proteins that have been found in plants. And we could find homologs of SCEP1 and SCEP2 in all clade of angiosperms. But what about the other protein we have in our screen? So we concentrate on another protein, which is SCEP3, which is another small alpha helix protein that we have in our data set. And this is the work of Marion. So in this SCEP3, we had a few univalents formed at in 5% to 10% of the cells we had univalents. So it's not as strong as SCEP1 and SCEP2. However, when we cross SCEP3 with SCEP1 and SCEP2, we had 50% of the cells in which we could find univalents, and not only one pair of univalents, but sometimes 2, 3, 4, or sometimes even 5 pairs of univalents. So the phenotype of SCEP1 or SCEP2 is increased, dramatically increased, when we have a double mutant with SCEP3. And what we found is that in SCEP3, ZIP1 is loaded normally. So SCEP3 has a very different role compared to SCEP1 and SCEP2. But what we could find is when we looked if SCEP1 could interact with SCEP3 and SCEP2 with SCEP3, we could see by AlphaFold or by these two hybrids that they could interact. SCEP2 interact with SCEP3, SCEP1 interacts with SCEP3. However, when we put the 3 together, we never found interaction with SCEP3 and SCEP1 and SCEP2. SCEP1 and SCEP2 interact more, always the interaction between the two is always preferred to the one with SCEP3. So we don't know at all. For the moment, it's an ongoing work, and we have raised antibodies. We are looking at the crossover number and position, and we are looking at the many epistases between protein involved in meiosis. And so what else? What else do we have in our list? We have also this protein that is called zygo1. So zygo1 had been shown to have a role in rice meiosis. It mediates bouquet formation in rice meiosis. In Arabidopsis, we have 3 zygo1. And Laurence and Mariana in our group are interested by the bouquet, by the dynamic of chromosome, and they had looked at many genes involved in the bouquet formation, and they had made mutant in the 2 other homologs of zygo1, and they hadn't found any role. So they are now studying this gene that is involved that could be involved in meiosis. What else? We have a gene that's called SCEP3. SCEP3 is a cache protein, and the cache protein interact with sun protein, and this how the chromosome move inside the nucleus with this sun cache complex. Again, Laurence and Mariana were very interested, so they had looked at SCEP1 and they didn't find any meiotic defect, SCEP2 and they didn't find any meiotic defect, and in SCEP4 they couldn't find any insertion. But when I looked at the expression of these 4 meiotic genes, the only one really looking very specific to meiosis was SCEP3. So they concentrate on SCEP3, and they could find that in SCEP3, the chromosomes were not moving anymore. So that the kind of studies they perform in live meiosis, they have a way to mark chromosome with the centromeric protein labeled with GFP. And in live meiosis, they follow the movement of centromeres within the nucleus. And that's the trajectory, for example, of centromer within a wild-type nucleus. But when there is a SCEP3 mutation, the centromer do not move anymore. So SCEP3 is required for chromosome movement, is required for telomere attachment to the nuclear envelope, and it's also involved in crossover formation. So it's also an ongoing work. So if I want to summarize what we found, we have this list of 80 candidate genes. We have screened 36 genes. We know that they have no meiotic defect. We have 8 with a clear meiotic defect. So SCEP1, SCEP2, SCEP3, SCEP3, zygo1. But we also have here this transcription factor found by Jan Anderson. And we also have here ATC21. ATC21 is a gene that has been identified by Stefan Heckman in this turbo ID proteomic assay. So he labeled the genes marking the lateral element. He looked at proteins that were marked by this proximity. And the strange thing is that between our list of 80 and his list, only one is in common, and it is this ATC21. And when he published his paper, we had this line in the greenhouse, and we confirmed that it has exactly the same phenotype than SCEP1 and SCEP2. So ATC21 is indeed involved in meiosis. And there is this gene C6. I'm not going to talk about it today because it's being studied by another group than our group. And we decided not to work on this one and to leave this gene to our to this other group. But we also have 6 other genes that have slight meiotic defects. So when I mean, you could consider that SCEP1 and SCEP2 have slight meiotic defects, but these 6 genes have even weaker defects. So instead of having 10 to 20% of cells having univalent, it's more 2 to 3% of the cells. So they are very hard to work with because the screening is very difficult. And we have to find a way to increase their phenotype to be able to study them. And we still have 31 lines to be screened. So can we decide how to choose between these 31 to be screened. So for that, Marion performed an AlphaFold interaction screen in which she put SCEP1, SCEP2, and SCEP3, and she looked at the interaction with all these 80 genes that we had, and she ranged them as this one having strong interaction and this one having weak interaction. That's the IPTM score. When it's close to 1, the interaction is very strong, and when we did around 0.2, it's very weak. So here are our genes. So SCEP1, SCEP2, SCEP3, and here you have this ATC21 gene, this C6 gene, and here our gene having a weak meiotic defect. So the one you have this sign here is just that we have screened and they have no meiotic defect. So we are going to concentrate on the gene on this end of the graph to see if we could find meiotic defects for these genes. So now what we are doing now, we are continuing our work on SCEP1, SCEP2, SCEP3 to try to understand better the dynamic of the localization with high resolution microscopy and live meiosis. We are continuing our AlphaFold interaction with other known meiotic proteins, and we try to find conditions to characterize the gene having a weak meiotic defect. And we still do the screening of the remaining candidates. And with that, I want to thank the people in our group. So the group is led by Mathilde Collomb here. The work has been done mainly by Nathalie Vrilinck and Marion Poche, but we got help of all the group here. And we collaborated with Raphaël Guérois for the structural studies. Delchip Finch-Sharif helped us to set up the bioinformatic analysis of the transcriptomic data. We collaborate with Raphaël Mercier for the STEM microscopy and the analysis of the genetic maps per genotyping by sequencing. And with that, I leave you here. And if you have questions, I'm happy to take them.
Any question? Yeah. Nahi? Joseph?
Thank you very much for a very interesting seminar. I have some 3 questions, and they are different type of questions. And the first one is related to the fact that you mentioned that in different organisms there is no homology in proteins and in transversal filament and central elements. But in this case, if there is no homology, how to explain that in different organisms we have the same organization of synaptonemal complex?
I think, okay, they don't have similarities in terms of sequence, but they have similarities in terms of structure. I told you that in most species, central element proteins are small coiled-coil proteins or alpha helix protein. So I think what is conserved is a structure. Okay. You need in the central element to have some small coiled-coil protein, probably to interact and stabilize the transverse filament, which is a zipper. And that's what I think is that what is conserved is a structure.
I see. Thank you very much. And the second question about the male meiosis, you mentioned that the number of crossovers in male meiosis is higher. What is the biological explanation of this?
I don't know if there is an explanation of that. In some species, there is no heterochiasmy. In other species, there are higher rate of crossover in one sex or another one. And at the extreme, there are species in which you have only recombination in one sex and not in the other one. What has never been found, I think, is the species in which you have this sexual reproduction without any recombination in both sexes. Mm-hmm. So I don't know if there is an explanation. I think you need a physical link. So you need a certain number of crossover, but after that, you need some crossover for diversity, but maybe you don't need much of crossover. I don't know. I think it's a cost. You have to balance the cost of having recombination and the cost of having too much recombination. So I think it depends from one species to another one.
I see. Yes, thank you. And the last question about this phylogenetic analysis of SCEP proteins here that you showed. It's very interesting that you found them in angiosperms, but and in lycopodialis, but not in gymnosperms. I don't see in these 3 monilophytes like ferns. What about ferns?
I think we tried to look at it and we could not find them.
Aha, okay. So simply no data available.
Yeah.
I see. Okay. It's really interesting results. Thank you very much.
Thank you.
Thank you very much for your very insightful seminar. I have 2 questions. One is related to what Dr. Dubrovsky asked you about the differences between male and female crossover frequency, but in this case, is if you found a phenotype when you have in the mutant, the SCEP1 mutant, you found that the number of crossovers are similar between female and male, right? So
yes,
there is a phenotype, particular phenotype in those mutants that may explain why the differences between these two.
Between ZIP1 and SCEP1. So in ZIP1, it's clear that I'm going to put it here. So in the wild type, we have clearly more crossover in male and female.
Right.
In ZIP1, it's the same.
Exactly. Yeah.
Both are at the same level. And in SCEP1, yes, indeed, it seems that we have heterochiasmy is not completely abolished. And to be honest, I really don't understand why. I think I think no, I don't understand. I have no clear explanation about that. I don't understand why we have heterochiasmy remaining, because if the synaptonemal complex is not formed, we should abolish heterochiasmy the same way in male and female.
Exactly. Yeah. Okay.
Yeah.
And the other question is, you said that the lack of homology is because, in fact, what the different proteins share is the structure, the secondary structure.
Yes.
So these proteins are, or at least one of these proteins may have another function in a different stage of the plant development, because apparently the fact that they are alpha helixes and they have this potential to interact with other proteins, they may interact with a different kind of proteins in another different process. So you know if they appear in a different kind of function or the process that is not necessarily meiosis?
They are really only expressed during meiosis, these 2 proteins. So I don't think for this one, I don't think that they have other roles. But as I show you here, I mean, we have many alpha helix protein here in our screen, and we really selected the one that were only expressed in meiosis. So for the one we choose, we think that they may be only expressed in this at this time of the development, I think.
That is the one that is homologous to, similar to topoisomerase. Did you say to topoisomerases or what? Tropomyosin, I'm sorry, tropomyosin.
Tropomyosin. It was called because in the database, when we looked at this protein, it was annotated as being a tropomyosis in the database. Yeah. It's just an homology of sequence because tropomyosin are alpha helix protein.
Right, yeah, I see.
So that was just a tropomyosin-like, as SCEP1 is annotated as being a myosin-like.
Okay, okay. Thank you so much.
Rosanna?
Hi, thanks for a very interesting talk. I'm not an expert in meiosis, but to me it looks like it could be a similarity between these proteins, the SCEP1, 2, with what is known in syntaxin in terms of vesicle trafficking, because in general there are several alpha helixes protein that interact to form a complex, and I have no idea whether your SCEP3 could behave like a regulator, or let's say that inhibiting or promoting the formation of this complex. I don't know whether it could be a possibility.
It's very interesting because effectively we think that SCEP3 is certainly doing something very different, and maybe we gave this name SCEP3 too rapidly, and SCEP3 is not at all involved in the central element, but is regulating completely different things during meiosis. So it could be another regulator. It's very interesting what you said, because the lateral element of the synaptonemal complex, I didn't want to go too much into details here, but you see here this structure, this lateral element structure here is made of proteins that are intertwining alpha helix to form that. So these alpha helix proteins are very interesting. They are really making complex that are very interesting to structure this synaptonemal complex.
So I don't see any questions from the broadcast, but I have one that is kind of intrigued me. From cytology, your mutants show some univalent, and in the genomic data, you don't see that you cannot detect a chiasmatic chromosome, or you cannot.
Oh yeah, we do.
Okay.
We do.
So can you detect that, and do they have particularity which chromosome do not recombine, or they are masked by the ones that normally do not recombine for parental segregation? I don't know.
So we did back cross, so we can detect if we. I mean, when you have in back cross, if you have only one crossover per bivalent, you'll have always two chromatids that will not recombine. So in your back cross, if you have zero crossover, it's going to be hard to see if it is due to the fact that there was no crossover, or that because of you got the chromatid that did not recombine in your bivalent. But we have an excess of zero, meaning that we certainly have chromatin that did not some crossover did not, some chromosome did not recombine, so we have an excess. So we know that I think what we found is that we found more short chromosome than long chromosome, so which we were expecting because they are smaller, and the probability not to have a crossover was higher for small chromosomes than for long chromosomes. Is that have you?
Yes.
Was I clear in my answer, or yes?
Yes.
But we have an excess of chromatid for zero crossover, so or genotypic map show that, or genotyping.
In your figure, it's misleading because you don't have any points that are on 0. That's the reason of my question.
Yes, that's true. But I have to. Where is that? It's further down. We have here. If I put it here, you see here, and not here. Maybe in SCEP1, SCEP2 we didn't have enough chromatids to have the power to see that. But I think we have. Maybe they don't appear here, but we have. I don't know why. I don't know why they don't appear. I will ask why, but I know we have.
Another question, in rice, they realize that SCEP1 can interact with HOP2, and HOP2 makes this kind of bias towards the repair on homologs. Have you tried with that scaffold to AlphaFold to see if HOP2 could be one of them?
No, we know it's very interesting. We haven't tried that. We tried hard. Nobody asked me if I could find any interaction with SCEP1, SCEP1 and SCEP2, and my answer is no. We have no interaction neither by AlphaFold or by these two hybrids. And the reason why is that SCEP1, the structure of SCEP1 is really poorly predicted by AlphaFold. AlphaFold is a fantastic tool, but sometimes it doesn't predict well the structure. And so we never could find. But that was more, the fact that we could not find this interaction by these two hybrids is still puzzling. There must be something else, and we don't detect this interaction.
Well, in other species,
but we are going to try with HOP2.
In other species, there is a lot of modification, like,
Yeah, I know.
Simulation, etc. So
yeah, I know, but it's still puzzling. I was expecting to find something.
Thank you so much, Christine.
Thank you very much for having me with you.